In this video, we're going to talk about the fungal phylum Zygomycota, which are also known as the conjugated fungi. The fungi in this phylum may be saprobes. Most of them are saprobes. Remember that saprobes are our decomposers. So they are breaking down dead matter into its smaller chemical components. Some Zygomycota are parasites, and they primarily parasitize insects. The thallus, remember the thallus is also known as the mycelium, consists of aseptate hyphae. So these are going to be the hyphae that are kind of underground or below the surface, that are essentially one big cell, but with several nuclei present. And this is because the cells go through mitosis, so the nuclei are copied and separate, but the cytoplasm does not separate. So cytokinesis does not happen at the end of mitosis. And these nuclei are haploid, which remember is 1N. And we'll talk about that more when we talk about the reproductive cycle of the Zygomycota. Now, these fungi do have a commercial role. They are actually used to make synthetic steroid hormones. And steroid hormones include things like the sex hormones, so testosterone, estrogen, progesterone, etc., and also cortisol. One example of Zygomycota that you might be familiar with is bread mold. So this mold that you see on bread is a form of Zygomycota. So Zygomycota primarily reproduce asexually, and this image here is actually kind of misleading because it doesn't even show this the production of these sporangiophores. But essentially what happens is that the mycelia develop these structures called sporangiophores, which release spores into the environment. And that fuzzy appearance that you see on bread mold, those are actually the sporangiospores that you are seeing. Now, when conditions are unfavorable, the fungus will switch over to sexual reproduction, and this sexual reproduction is called conjugation. And it follows kind of the same basic steps of fungal reproduction that you learned about in the fungal reproduction video, but there are some specific structures that form in this particular type of fungi that we're going to talk about here. So let's talk more about the sexual reproduction that occurs in Zygomycota. So first of all, Zygomycota have heterothallic plasmagamy. So hetero means other or different. Thallus, remember we're talking about the mycelia. And plasmagamy is the first step in fungal sexual reproduction. And during plasmagamy, the cell membranes and cell walls in the cytoplasm of two cells fuse together, but the nuclei do not fuse together during this stage. So heterothallic plasmagamy essentially means that we have two different fungi. So this is going to be one fungus with the mycelium made up of these hyphae. And then we're going to have a separate fungus with its own mycelium. Now, each one of these is going to be a different mating type. So we'll just say that red here is the positive mating type and blue is the negative mating type. Now, what happens is that these fungi give off pheromones, which are these chemical messengers. And essentially, it causes these hyphae to start to grow towards each other. And note that these are aseptate hyphae. So that means that these are long cells that have multiple haploid nuclei in them. And you can kind of see this in the picture here, where you can see these mycelia made up of these aseptate hyphae. So notice that there are not these divisions in between the cells. And as these hyphae grow towards each other, they start to form a gametangia. So the red one will form a gametangia, and the blue one will also form a gametangia. And these are just going to be little kind of bumps or extensions coming off of the hyphae, and they're coming together towards each other. And you can see this happening right here in this picture here. So the gametangia from the two separate hyphae of the separate mycelia are basically extending out towards each other. Now, ultimately, what happens is that these gametangia are going to fuse during plasmagamy. So this kind of area here fuses to form a zygosporangium, and that is being shown right here. And this zygosporangium, and I'll kind of draw it over here to make it a little bit bigger, is going to be a single cell, but it's going to have multiple haploid nuclei in it. So because the hypha are aseptate, it means that when these gametangia form, there can be multiple haploid nuclei in each of the gametangia. And then when plasmagamy occurs, it forms a cell with multiple haploid nuclei, and some of the haploid nuclei are from the positive or red mating type, and some are from the negative or blue mating type. Now, during karyogamy, which is our second step here, those haploid nuclei are going to merge, and so this cell is ultimately going to become a cell with multiple diploid nuclei. And I am drawing these diploid nuclei in purple because when you mix blue and red together, you get purple. But remember that when we're talking about diploid nuclei, we're talking about paired chromosomes. So inside these membrane-bound nuclei, you would see these paired chromosomes of red and blue in each one of these here. And on this picture here, we can see that right here. And this structure is known as a zygospore. So to recap, this cell here is a zygosporangium. And this one with the diploid nuclei is a zygospore. Now the zygospore is going to go through meiosis. And remember that with meiosis, the paired chromosomes essentially get shuffled and then kind of dealt out into the gametes. So we end up with a different combination of chromosomes. And in doing so, we end up creating these spores that might have a combination of red and or blue chromosomes. So this is how we have increased our genetic diversity by taking these two separate mycelia, forming the zygosporangium, letting those nuclei fuse during karyogamy. And then in meiosis, those nuclei are then going to separate back into haploid nuclei, but now they have a new kind of different combination of chromosomes than what they started with. Now, one thing to note is that these zygospores have a thick kind of coating around them, and it allows them to survive in adverse conditions. So these zygospores can actually remain dormant until the environmental conditions improve to a point where it's favorable for the fungus to germinate. So during germination, these haploid spores are going to be released from the sporangium. And if they find a suitable place to land, then they can germinate and then make a new mycelium. And they can do that either asexually or they can then go through sexual reproduction again.